Hello guys, I am Shubham from Fluid Tutorials. This video is about transduction. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. So let's get started with the video. So what is transduction? Same here, like conjugation and transformation. Transfer of genetic material takes place into the bacteria, but the transfer takes place through bacteriophage. Okay, or we can simply say phage, right? Transfer of genetic material in bacteria, right? So all this conjugation, transduction, transformation, genetic material is getting transferred into the bacteria, right? So this transfer in transduction, the transfer occurs through bacteriophage. So what is bacteriophage? Bacteriophage are the bacterial viruses that cause infections in bacteria, right? These are also known as phages. Okay, phages, right? So these are the vectors. We can say phage vectors that will transfer the genetic material from one bacteria to the another bacteria, right? So life cycle of phage is very important before we discuss about transduction. So we will start with life cycle of phage. So one is the lytic cycle. A lytic cycle. Lytic cycle means there will be lysis of the cell. Okay, bacterial cell. The virus will grow inside it, and virus will take all the machinery of the bacterial cell, produce its copies inside the bacterial cell, and then cell will lysed. Okay, there will be cell lysis, and the virus particle will release inside into the environment, and then it will infect the another cell. Right. In the second life cycle, lysogeny, lysogenic cycle. Lysogenic cycle means what? The virus particle inserts its genome, right? And that genome will get integrated into the bacterial chromosome, right? So the virus genome will get integrated into the bacterial genome and it will stay inside it. It will replicate with the bacterial chromosome, right? But when the condition is favorable, the lytic cycle will switch on. Okay, so it will come out of the bacterial chromosome after appropriate conditions, right? It comes out from the bacterial chromosome and then again same lysis occurs, virus will infect the other cell, bacterial cell, right? So we will discuss this. Now we will start with exact transduction. So there are types of transduction. Types of transduction. First is generalized transduction. Generalized transduction, and second is specialized. Specialized transduction. So in generalized, any genes can get transferred. Okay, any genes. And specialized means specific genes will get transferred. Only specific genes. And in generalized, any genes can be picked up by the phage. Right? So we'll see here with the pictures. Actually, what we can say here, generalized is the related to lytic cycle. Generalized related to lytic cycle. Okay. And specialized related to lysogeny. Lysogenic cycle. Okay. Also, we can say this is related to temperate phage. Temperate phages. What is temperate phase? Temperate phase are the phases that have the option, okay, have the option to remain in the lysogenic cycle and initiate lysis when the conditions are appropriate. Means these phages can have both the types of life cycle. Right? Temperate phase. I will write here temperate phase have choice between lytic cycle and lysogenic cycle. Right? So this is a temperate phase. Here the example is lambda. Example is lambda. And lytic cycle followed by T2 and T4 phases. So that switch, why it has the option to switch from lysogenic cycle to lytic cycle? How we can switch? There is a protein known as repressor protein, okay, which inhibits the lytic cycle. But when that protein is degraded, the lytic cycle is switched on. Okay, so that protein known as repressor protein. It is a product of gene inside the present inside the temperate phase. So now let's see the exact process with the help of pictures. How does generalized transduction occurs first? See, generalized transduction between strains of E. coli. So here is a phage, right? Inserted its genome inside it. When the genome is inserted inside the bacteria, what happens? The bacterial chromosome is degraded. See here, fragments of bacterial chromosome degraded. Okay, right? See, it is written phage reproduction by lytic cycle along with breakage of bacterial DNA into fragments. Means the genome is started to express the virus particles. It will produce its particles inside a bacterial cell. The whole process of replication is inside the is controlled by the phage. In these cases, when the genome is inserted, all control is in the hands of phage, and thus the bacterial chromosome is degraded. After this, see all the particles will be created of virus particles, right? Now, normal phages. Okay, normal phages. Normal phages means these are normal with its own genome. We can see pink genome is the phage genome. So those are inserted inside the virus particle. Now, here what we can see transducing phase. What are transducing phages? So these are the phages with bacterial chromosome. Okay, bacterial genes are inserted inside this. Bacterial chromosomes are inserted. So these are known as transducing phages. Now there will be lysis. Cell lysis. After cell lysis, the particles are released. Right? Phage particles are released. So these are non-transducing phage with its own genome, right? With its own genome non-transducing, and these are transducing with the genome of bacterial chromosome. And then when this transducing phages, see, when this transducing phages will infect the bacteria, other bacteria, second bacteria. Suppose we say this is the first bacteria and this is the second bacteria. Okay, so this is the first bacteria getting infected with this phage. Now, this phage is getting developed inside the first bacteria, and then after lysis from the first bacteria, it picks up the bacterial genes and infecting the second bacteria, right? Then second bacteria, what happens? The phage inserted its genome here. Now there will be double crossover event. Okay, double crossover because it is homologous. And after double crossover, what happens? The chromosome is exchanged. See, right? This is the thing. So we can see here, this is a stable transduced bacterium A plus transductant produced by recombination, and this linear fragment degraded by cellular nucleases. So this is all about general transduction. Now, specialized transduction, same things happen, but this is very specific. We can see here, and this is seen in the lambda. Specialized transduction by bacteriophage lambda. What happens? Production of lysogen by crossing over between circular bacterial chromosome and circularized phage chromosome. So this is the genome of phage having the site specific site ATT. ATT is the attachment site. And 880 lambda, see vector chromosome is having 880 lambda, means attachment site for lambda. So that is cross between these two sites, and then it will get integrated, right? See, it's getting integrated, right? So this integrated will replicate inside the bacterial genome for some time, right? And when we induce the lytic cycle, if we want to induce that for genetic mapping or to understand the gene order, if we are studying for that purpose, what we'll do, we will induce the lytic cycle. For this induction of lytic cycle, what happens then after induction, this integrated lambda chromosome will come out. See, will come out. What will happen? Production of initial low frequency transducing lysate. When induction of lysogenic bacterium causes outlooping. Outlooping means excision. When excision takes place of this lambda chromosome, it will occur accurately or sometimes inaccurately. Right. See, this is normal outlooping produces normal lambda phases. Right. Normal phases are produced, but a rare abnormal outlooping produces transducing lambda D gal plus phases. Okay. Lambda D gal plus means defective. Right. D stands for defective, and it has taken some parts of bacterial chromosome gal plus C. Lambda D phase chromosome. So defective. Now this after lytic cycle is induced now, right? Then after releasing out of the cell, the genome, okay, genome is ready. Now there are some particles of normal phase chromosome. Phase is lambda here, right? And this is lambda D defective, gal plus phase chromosome, which has taken gal plus is a bacterial gene, right? So after release, after lytic cycle is completed, this will get released and this will then infect the other bacteria. See, this is written. Transduction of gal plus bacteria, sorry, gal bacteria, because it is not plus, by initial lysate, right, consisting of lambda and lambda D gal plus phases. This phase will be inserting its genome into the gal bacteria. Bacteria is mutant for gal. In that, there are two cases, right? The first one is what? If both lambda and lambda D gal plus integrates an unstable transduction, a double lysogen results. Right, means double lysogen results. Double lysogen means lambda phage chromosome, right, as well as lambda D gal plus chromosome. Both the chromosomes are getting inserted inside the bacteria, right? So after this, what happens? Induction produces about equal numbers of lambda D gal plus and normal lambda phages, a high frequency transducing lysate. So this is known as high frequency lysate. If this is high frequency lysate, then why the previously it was known low frequency lysate? I will just explain. Why this is known as low frequency lysate, reproducing lysates? Because normally the excision or we can say outlooping is accurate. But sometimes the outlooping is rare, not accurate. Okay, this is in rare conditions. So thus it is known as low frequency transducing lysate, right? Because this condition, outlooping occurring inaccurate, is very rare. Normally, it is accurate. So thus, it is known as low frequency (LFT). And this is high frequency because both the genomes are getting inserted inside the bacterial chromosome, lambda phage, right, as well as lambda D, defective one. Both the chromosomes are getting inserted. Now, in the second part, what happens? In the second case, this is a stable transduction produced by recombination, right? How so? Here only lambda D gal plus will infect. Okay, see lambda D gal plus phage chromosome. Only that will infect the bacterial chromosome near the gal region, right? Double crossover. Thus it will exchange the gal plus gene only. Okay, see gal plus gene only it is exchanged. Lambda D gal plus phage chromosome. So it is unable to replicate because not all genes are present. Right, gal is exchanged. Okay, so this is inactive, unable to replicate. So this is stable transduction. So all this, these are specialized. Okay, I will just summarize the specialized one. So this is the first bacterial cell, right? Bacterial cell having its own genome, double-stranded genome. Okay, then what happens? This is a temperate phase. Infecting a genome, infecting the bacterial cell. This is lambda 1, temperate phase. Now what happens after it's inserted its DNA, it gets integrated inside this. Okay, this genome of phage gets integrated inside the bacterial genome. So this will replicate with the bacterial genome as and when replication takes place. I will just draw here in black and other one with red. So see, this is integrated. Now this is in the what we can say repressor is active. A repressor is what? Inhibitor of the lytic cycle. That is active. So as it is active, it will stay in this integrated state. Now we will give some of a treatment that it will inactivate the inactivate a repressor. What is repressor? Inhibitor of the lytic cycle. If we inactivate that, then what happens? Bacterial chromosome is there and phage genome is coming out of the bacterial chromosome. Like this, it will come out and initiate the lytic cycle. Then this it will repeat the same as we saw in the general transduction. The original bacterial chromosome will be degraded. Right? All the control will be in the hands of this phage genome. Right? So all the virus particles will be produced inside this. Okay, and then the virus will be released by the cell itself. Right? So whole this process is in the specialized transduction. So same here is also a term known as co-transduction, or we can say co-transduced, means when the two genes are close to each other, those genes are picked up by the single bacteriophage or virus, right? And then this both the genes get transferred at a single time, right? So this is known as co-transduction. And with the help of this, we can understand the gene order as well. Gene order, or we can say gene distance. Basically, gene mapping, genetic mapping in bacteria. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Thank you guys for watching this video. If you like it, please share it. Don't forget to subscribe and press the bell icon to get notified whenever the new video is out. Also follow us on all our social media pages, and you can join us on Telegram and stay connected for the latest updates. All links mentioned in the description section.